Okay, so hi, my name is Vineet Reddy, and today I'm going to talk about some of the stuff I do in the lab studying autism. So these are two pictures of me. I'm 20 years old. I was raised in the suburbs of North Atlanta. I'm currently a neuroscience major at the Honors College of Florida Atlantic University, which is located in a separate campus in Jupiter, Florida. And I work in McLean Bolton's lab at the Max Planck Florida Institute of Neuroscience, where I've worked since freshman year of college. So, why do we study autism? Autism is a very interesting disease that affects millions of children across the world. So, around 1 in 70 children across the world are affected by autism spectrum disorder. And interestingly, autism spectrum disorder is often associated with other disorders such as epilepsy and other disorders that are related to uh, different functioning of neural circuits, or essentially neurons communicating within loops. An interesting figure related to this is that around 30% of children who have autism develop epilepsy later in life, which tells us something about how neurons are acting differently in children with autism and children with autism and epilepsy. And it's also an interesting disorder to study from a fundamental neuroscience perspective because it tells us how our brain is wired and how people with autism, how their neurons connect differently. So this is kind of a general workflow as to how our lab studies autism. So the first part of the process is we have to use an animal model because we have to study this disorder within an animal model to understand more information about how the neurons connect and how the animal behaves. So first, we have to create a mouse model. And to do this, we look at genes in humans that are different or mutated, um, in which specific amino acids within the DNA are changed within humans. And then we can look at how these genes are different in humans and take that and look at a mouse genome and change the same gene within a mouse to understand how autism can affect humans in the same way that autism can affect a mouse. So in my case, I study this gene called neuroligin 3, and what we do is we go to position 451 of that gene, and we change an arginine to a cysteine. And so this gene mutation is the same gene mutation that was found in a pair of twins in humans. And so we're taking a mouse gene that's implicated in autism and changing it. And then what we do is once we have this gene mutation within a mouse model, potentially implicated within autism, we can analyze how that gene mutation affects the way that the mouse behaves. So either the way the mouse behaves within a setting by itself, or the way the mouse behaves in front of other mice as well. And the third process in this workflow is known as electrophysiology. And so this is an interesting method in which we kind of tap into the phone line of the neuron, and we kind of eavesdrop into how each neuron talks to each other, either right next to each other or within circuits in between brain areas. And lastly, we kind of combined what we gleaned from the behavior that we saw as to how the mouse acts and the electrophysiological information, so how the neuron sends signals to other neurons, to see whether what we're looking at, what gene we're looking at, is potentially a target in the future for future therapies to affect autism in the future. So this is an example of one of the behavioral tests that we run. And so this is called an open field test. And it's a classic neuroscience behavioral test. So in this test, what we do is we place a normal mouse in the mouse box, and then we take a mouse with the gene mutation and we put it in a separate box. And what we do is we analyze what the mouse is doing as it's running around. So is a mouse mostly in the center? Is a mouse mostly at the edges? Is a mouse moving around a lot? Is a mouse not moving around a lot? We can quantify many of the different behaviors that the mouse is doing while it's running around in this cage. And what we can see is we can see how these behaviors are differently affected within the gene mutation mouse. So for example, if the mouse is mostly in the center for a mouse without the gene mutation, and the mouse is mostly at the edges in the mouse with the gene mutation, that can tell us a lot of things about how this gene is affecting the way that the mouse can interact with its environment and with the context surrounding it. Another method that is very prominent in neuroscience and a method that I personally work on in the lab is known as electrophysiology. And this is a way for us to look at how a single neuron acts within a brain slice, and we can see what currents the neuron are sending out to other neurons right next to it. And so on the left is my workstation, and it looks very, very complicated, but I'll break it down for you. So the big white block right there is my very, very expensive microscope. And under that is the lens objective. And under the lens objective, in that little hole that you see right there, is where we place a brain slice. And what we do is once we place a brain slice in there, we run a solution, the same cerebrospinal fluid that runs in your own brain and spine, we make an artificial version of it to allow the brain slice to exist in somewhat the same environment that it would exist in a mouse itself. And so we run artificial cerebrospinal fluid over the brain slice. And then what we do is on the right, you see a tiny, tiny glass needle. We can take the glass needle and we can poke neurons within the slice. And then we can establish a seal between the glass needle and the actual cell to measure how the cell itself is responding. And so this is what I would see in the microscope itself. And so on the left is the same glass needle that I was just talking about. Sometimes we call it a pipette. And on the right is an actual single neuronal cell. And what we're doing here is we're establishing a circuit. So the pipette is actually able to record from what the neuron is saying. And so we jam the pipette into the neuron. We form a seal with the neuron membrane. And this allows us to see exactly what the neuron is saying to other neurons right next to it. And so this is just a picture of me. And what I'm doing here is I'm essentially just working on the rig to patch onto one of the cells and see how the cell is responding. And what we can do is we can ask the cell different questions. We can stimulate different brain areas and see how the cell responds. Or we can even control how the cell is responding itself. We can change the way the voltage at which we hold the cell to either make it fire or not fire, and see how other cells respond as well. So these are the main methods that our lab uses, but now I kind of want to switch to what I do in the lab myself. And so my project regards how inhibitory interneurons talk to each other differently in people with autism. So there's a lot of loaded terminology in here, so let me break it down. So what is an inhibitory interneuron? So an inhibitory neuron is a neuron that ramps down a cell signal. So let's say we have 3 neurons. We have neuron 1 on the left, neuron 2 in the middle, neuron 3 in the right. If neuron 1 fires at neuron 2, and then neuron 2 fires at neuron 3, if neuron 2 fires such that neuron 3 is less likely to pass on a signal, then it's defined as an inhibitory neuron as opposed to an excitatory neuron. And so what is an interneuron? An interneuron is specifically a neuron that is between other neurons. So because neuron 3 is between neuron 1 and neuron 3, it's defined as an inhibitory interneuron. And so our hypothesis is that these inhibitory interneurons interact differently with other neurons in people with autism. And so we kind of looked into it. So what I did is I used this method of electrophysiology to patch on to inhibitory interneurons within this brain area called the lateral amygdala, which is responsible for inputs into the amygdala. And the amygdala, if you don't know, is important for a lot of emotional regulation. And so what I found is that there is no significant difference in the mini excitatory currents that these inhibitory neurons were sending out. So Phlox is the group of mice with the gene mutation, and control is the mice without the gene mutation. And the blue bars represent the amplitude of the current that is firing from the inhibitory neuron and the frequency of the current that is fired from the inhibitory interneuron. And so amplitude and frequency tell us different things about how the cells are firing. But as you can see, the flox animals, the cells in the flox animals fired at a lower rate, so at a lower frequency, and had a less amplitude of firing, but it wasn't statistically significant. But as you can see, there's a general trend. And so that kind of clued us in to what the next part of our project is. And so the next part of our project is essentially going from inhibitory neurons into specific types of inhibitory interneurons to see if this trend is more drastic and more significantly different in specific subtypes of inhibitory interneurons within the lateral amygdala and also other brain areas like the basal amygdala and the central amygdala. So the last thing I kind of want to talk about, other than my science, is about my experience in the lab. So I joined my lab in freshman year of college, and it has been the single most exciting and rewarding experience that I could have possibly done. And one of the main things that I learned from working within a laboratory setting is the ability to problem solve while working as a team. So there's multiple things that can go wrong when you're using an electrophysiology rig. And most of the time you have no idea how the electrophysiology rig is messed up. And so problem solving comes in when you're able to go in methodically and check every little detail and use your own experience as an electrophysiologist to figure out by yourself what is going on. And if you're able to figure this out, it's very, very rewarding and you're able to progress science. But you also need to work as a team in order to problem solve. Many times, as I said, the rig is messed up and you have no idea what's going on. You have to go to other individuals in order to work with them, to figure out, to put your heads together and work as a team to progress science and work at the forefront of current technology in order to figure out what's going wrong and do your experiments for the day. And not just in terms of messing up the ePhys rig, but when you're working on a project and you're trying to find out more knowledge, the more heads is the better. And some of the most important lessons I learned is that even if you're very, very smart, you have to work with others in order to progress a project and do better science. And so that's kind of my experience within the Bolton Lab. And I hope you enjoyed our presentation today. You can follow us at our Instagram handle at EngageFL, and you can shoot us an email at fauengage@gmail.com or visit our website if you have more questions.
Good morning, everyone. My name is Jose Delgado again, and today I'm going to be talking a little bit about inhibitory circuits, neural circuits. Does anyone know what an inhibitory neuron is before we get started? I know Vineet talked about it a little bit. I can't see the screen, so if Matthew or Vineet or Miss Lynn, if you guys can tell someone to answer.
Anybody remember what he said about inhibitory neurons? Don't be shy.
All right, no worries. I'll go ahead and touch on them a little bit as well later on. But for now, I just want to talk a little bit about my experience and who I am. So, as I mentioned before, I was actually in the medical academy since 6th grade, and I was part of the FAU Biomedical Sciences Academy. So, as you can see, this right here, that's me holding a real human brain in FAU's cadaver lab. And this is me in 6th grade as well, dissecting what I believe was a rat. So it was a really fun experience. And since I was very young, I always loved medicine. And I just loved neuroscience and the human body as a whole, and I thought it was very interesting. And I continued the medical program from Lake Worth Middle to Lake Worth High School, where I had Miss Lynn and Dr. Ez, Dr. Garcia, and everyone there. And throughout this journey, I started learning more about the nervous system. And to me, it was really interesting because it's complicated but beautiful at the same time, you can say. And I heard about Max Planck. And Max Planck Institute is basically a research institute, and there's various institutes, especially in Germany. There's only one in North America, and it's in Jupiter. And each institute, they do research on different stuff. So maybe there's a Max Planck Institute of Sun Power or Mechanics. The one in Jupiter is Max Planck Institute of Neuroscience or Neuroscience. And I learned that they actually offer an internship there. And I even asked Mr. to write me a letter of recommendation for their internship. And it was a very heavy internship to try and get into because there were hundreds of applicants and they only chose 6 students. But luckily, I got accepted. So as you can see, I actually worked with my mentor, Dr. Andre Steinig, in the Taniguchi lab, which I will talk to you a little bit later. And these are also my fellow interns. And this is Ilaria Drago, she's amazing. And overall, this internship was really amazing, especially because I come from Lakewood High, and we all know that Lakewood High is a Title I school. And I am personally very competitive. And for me, I really want to get this internship, not only for myself, but to prove that, hey, you know, I'm able to compete with students from Suncoast or these top name high schools, whereas our high school really isn't that big. Matter of fact, it has a negative connotation attached to it. So I was really proud of myself to be able to do this. And this just goes to show that it doesn't matter what school we come from, but we are able to compete with all these other students. I mean, this student right here, he goes to Suncoast, she goes to Jupiter High, they go to a private school, and I come from Lakewood High, but I was still able to compete with these students, and that's something that every one of us can do. Now, during this internship, I did some really cool things in my opinion. So for instance, this is a picture of, can anyone guess? Anyone know what this is a picture of?
You're they're saying a plant cell or a cell.
Yeah, so a cell. So right here we have a cell, and I actually took this picture. And this is a picture that you might see on the textbook or on a professional website, but with Max Planck's resources, I was able to take this picture. And I can say that it's my picture. I just found that really cool. I also did some other experiments such as electrophoresis. So it's basically, I like to think of it as racing DNA. So obviously I don't have a picture of the actual procedure because we have to use a certain machine, but this is a picture of the process. And these two things are basically strands of DNA or RNA. And I just have them racing each other through a gel. So on this end, it's, if I remember correctly, negatively charged, and this end it's positively charged, or it might be the other way around. And basically the DNA strands or RNA strands are just racing through a gel. And it's a really cool experiment, and that's what I was able to do. But onto the more juicy stuff and what we're here to present. Well, I did an experiment with inhibitory neural circuits. So the way that a neuron actually passes information is through an action potential. And I like to think of it as a wave. It's an impulse that travels through the neuron from one end to the other. And from that end, it travels to the other neuron. So I guess you can think of it as, you guys call it telephone, I believe the game is called, where it's like you tell a person next to you a message and they have to relay that message across everyone. So that's one way to think about it. Now, an inhibitory neural circuit or neuron is a neuron that actually prevents or decreases this action potential from occurring. So you can think of it as, in this game of telephone, maybe you keep on passing this message across your partners, but there's one person in the middle who just decides to not spread the message. So he cuts that off. That's one way to think about it. Now, an example of an inhibitory neuron is the chandelier cell. And it's this very big green structure that you see here. And you can see why it's called a chandelier cell, because it looks like a chandelier and it has a lot of projections. And right here, this is called the axon initial segment. Now that was on the paper that we gave you guys. So does anyone know what an axon initial segment is?
No, I didn't get it. I didn't go over that.
No worries. So an axon initial segment is the area on a nerve where the action potential starts, or where this wave of impulse starts. So this is the area that the chandelier cell actually innervates or connects to another neuron. So right here we have a graphic picture. So we have a pyramidal neuron that's trying to relay a message to other neurons, and the chandelier cells actually connects this pyramidal neuron and stops it from sending these messages because it stops the action potential. So again, the goal of this inhibitory neuron is to stop these signals from occurring. Now, why do we want to study these chandelier cells? Well, first off, there's not much known about them, so we really have to get to know them more in order to study the brain as a whole even more. So we just have to do more research and background research on them just for a general overview. But not only that, but it's also to study diseases. So inhibitory neurons, they actually prevent hyperexcitability of neurons. An example of hyperexcitability is seizures. So when the seizure occurs, it's because all your neurons are firing and they're going crazy. Inhibitory neurons can actually prevent that from occurring. Not only that, but also psych diseases or illnesses, because bipolar disorder or schizophrenia is also due to these neurons firing all the time without being regulated. So we have to study them in order to better understand these areas. So how did I study these neurons? Well, my experiment was called the in situ hybridization of acetylcholine receptors in cortical chandelier cells. Now, that's a big mouthful, and it took me a long time for me to actually understand what this meant. But during this Max Planck internship, I worked in the Taniguchi lab, and their focus was to study inhibitory neurons. And my mentor, Andrzej Steinek, was specifically studying these chandelier cells. And as you saw in the picture, these chandelier cells, they have a lot of projections. You know, they branch out and connect to a lot of neurons. But he asked himself, what causes these neurons to actually grow and branch out? Well, he learned that acetylcholine, which is a neurotransmitter, actually causes them to branch out. But in order for these neurons to actually be affected by this neurotransmitter, they have to have receptors, okay? So another way of thinking of it is like two people, they have two phones, right? If you're trying to get a hold of someone or send them a message through your phone, they have to have a phone as well to receive the message. So same thing. The neuron, in order to actually be affected by this neurotransmitter, they have to have a receiver, something that receives their stimulus. But there are different types of receptors. There could be alpha 1, alpha 2, alpha 3, beta 1, beta 2, beta 3, and so on and so forth. So my experiment was to find out which type of receptor is most dominant within these neurons. In order to do this, I did a procedure called in situ hybridization. Now, in this neuron, they have a specific, or this receptor has a specific RNA or genetic sequence. And does anyone know what a complementary sequence is? I'm not able to see the chat or anything, so if Annie or Matt or Ms. Lynn, anyone can choose someone.
I don't think they're gonna know, Jose.
All right. So we have a genetic sequence, right? And a complementary sequence is one that just matches it. So for instance, you can think of it as a jigsaw puzzle. We have a genetic sequence, which is a jigsaw puzzle, and we have another puzzle that directly matches it, or fits right next to it. So I have my target RNA, and I have to create a complementary sequence that attaches to this RNA strand. And that's represented by this green structure. This green structure also has this bright red dot, and that F stands for fluorescence. So basically, if I look under a microscope to observe these probes, that I call them, I'm going to be able to see a bright red light. So again, another way to think about it is So I have my target RNA, which is going to be the genetic sequence of these receptors, and I connect them to a jigsaw puzzle piece, right, that attaches to them. And this other puzzle piece has a bright red light, it's fluorescent. So if I look under a microscope and I see this big red color, that means that my target RNA is there. So this procedure actually took a long time, and the internship was for 10 weeks and 6 weeks, excuse me. And this in situ hybridization part actually took 1 week just to do it one time. So it was a hefty process. But what was this process? Well, first I had to actually design the insert. So I had to create the probe, the thing that's going to attach to my genetic sequence. So what I did was that I designed an insert, and this insert was the same genetic sequence as the receptor, and I'm doing it for a beta-2 receptor. And I attached it to this Caterion 12 genetic sequence. So basically now I have a complete genetic sequence, and this green aspect is the same genetic sequence of that of the receptor. Now what I did is that I actually replicated this whole genetic sequence through bacteria colonies. So I had dozens, and I had hundreds of bacteria in a little dish, all that had this genetic sequence. Now again, this is the same genetic sequence as the receptor, but I want the complementary strand. So I added a T3 promoter, and what it does is it goes through this green strand and it creates the complementary strand. So once I did that, I had my probe. And then I did the actual in situ to actually combine my target genetic sequence and this probe. And then I looked at it under a microscope. And this was my final picture. So all of these bright red dots or signals are actually my probes. So all of these areas are where beta-2 receptors are located. Okay. So through this, I was able to find out that the nicotinic acetylcholine receptor type beta-2 is most dominant in the chandelier cells. And once I finished with that experiment, I actually had to present in front of the Brain Trust Institute. It's a bunch of very important people who actually funded the internship. And this was at the end of my internship. That's what I had to do. So overall, this was a very fun and a very intriguing time. I would say it was one of the best summers I've ever had, because I was able to do something really cool and I was able to learn a lot. But I think what's most important here is what I mentioned before, that this all started from a simple biomedical sciences academy from Lakewood Middle and Lakewood High. And for me, I'm very passionate about education. So just knowing that, hey, I was able to compete with people from Suncoast to achieve this internship, it just proves that any one of us can be able to do research and go to these top tier internships or opportunities. And we just have to look for them and take advantage of them, honestly. And with that, I thank you guys for listening to me, and I'm open to any questions if you guys have any.
Hello, everyone. My name is Matthew Pacheco. Thank you so much again for letting us borrow your time this afternoon. Today, I'll be talking about my research, so a brief introduction of myself. So I go by Matt. I'm 20 years old. I was raised in South Florida from the age of 10 until now. Before that, I was actually living in Ecuador with my family. And I'm also a cellular neuroscience major at the Honors College of FAU, like I previously mentioned. I currently work in Dr. Stern's lab at MPFI, but I will be talking about the work that I did at the molecular core, and I'll explain what that is in just a bit. But first, I'd just like to just talk a little bit about myself before I go into the science, because I know there are a lot of big words and just this vocabulary that you guys might not have been introduced to yet, or maybe it's just a matter of, until you get to college. I know when we present, we talk about all these things, but there was a certain point in time when we didn't understand these things, and it took a lot of learning and working and studying to grasp these concepts and learn about the science that we work with. So yeah, I'm a first generation student. My parents are immigrants. We came to the US when I was 10 years old. And this caused me to not it caused some uncertainty on what I wanted to do later on in life. I didn't really know how to get involved with clubs or any extracurricular activities that might enhance my opportunity to go into science or medicine. Luckily, West Boca High School, like many other high schools in Palm Beach County, have medical science academies. So I did enroll, and I was there all 4 years. And while I wasn't, the best student throughout high school, it did inspire me, and I did always know that I wanted to be a doctor. And a program such as the medical academies in Palm Beach County was very beneficial to my growth and just self experience going into college. So here are pictures of myself. On the right, you'll see I'm in the lab. I think one of my experiments worked, so I was happy and I took a selfie. In the middle, so another fact about myself, I'm also an emergency medical technician. So I worked in an ambulance for 2 years, responding to emergency calls and transporting all sorts of different patients. In the middle, right here, I'm in the Department of Homeland Security office at Fort Lauderdale Airport. I was doing a COVID operation with them when it first broke out back in March. So I was helping with, passengers coming in internationally and all that sort of stuff. So that's just kind of a background on what I do. And, I will say, I always knew, like I said before, I always knew I wanted to become a doctor. I just didn't know how. And that's why we're here to talk to you guys. If you guys want to go into research or medicine or anything involving, research, it doesn't only have to be science. There's research in the humanities, social sciences, all that. It doesn't have to be, like a hard science, like biology, neuroscience, anything like that. So that's why we're interested in engaging in these conversations with everyone. So I'll go ahead and talk about my science. So what is molecular biology? So when you hear the word molecule, you might think of like a very small, maybe like an atom or a very small structure. And that's right. A molecule is a very small structure, that has a specific function. So the branch of science that's called molecular biology deals with the basic components of life. So molecules such as, DNA, mitochondria, all these terms you probably heard before in your basic biology class. So these components of life are very beneficial to study, because that way we can learn from them and we can learn how to manipulate them. So when you learn how to manipulate something, then you're able to use it to your own advantage, right? So that's why studying DNA, proteins, and all these cellular molecules is super important, especially for neuroscience, because the overall goal of neuroscience is to dig into the brain and learn different facts and ways to go about solving some neurological disorders, maybe, or there might be some mechanisms like what Vineet was talking about earlier. He was talking about autism spectrum disorder. So there are just different, there's so many things in neuroscience, and at the basis of it, it's just molecular biology, because that's how we observe things and we learn things. So I'll go ahead and talk about my specific project and how I use molecular biology to, use this method in neuroscience. So like I said, here's the Max Planck Florida Institute for Neuroscience. It's in Jupiter, Florida. And my project involved the use of these things called viral vectors to image neurons via fluorescent proteins. So I'll explain what that means. So a neuron obviously is a brain cell, and a viral vector, I'll explain what that is. So we use viruses in the lab. They're non-pathogenic, which means they're not harmful. They don't make you sick. They're just viruses that we use to our own advantage. So we'll actually make homegrown viruses in the lab to express a green protein inside of brain cells to then make the brain cell glow green. And there's not just green, there's multiple other colors that you can make a brain cell glow. So it's actually pretty cool because a virus is actually just a protein capsule. So a protein is just a capsule, right, for this virus. And then there's genetic material inside of this capsule. Now there's different forms of viruses, but the viruses that we use, they just go inside a cell. They inject their own genetic material into the cell, and then they use the cell's replication mechanisms to replicate that own genetic material that the virus introduced. So it actually hijacks, the machinery that it uses to replicate inside the cell to then make more virus. And, that's actually how coronavirus works and other viruses that we know about. And the replication is what makes people, ill. But in this case, we are just expressing fluorescent, glowing brain cells. So it's totally harmless. So here I am. I don't have a picture with my lab because although I've been working there for like a year and a half almost, I just never got the opportunity to take a picture because of COVID. So here's my picture on the website. So yeah, here's DNA, right? You guys are all familiar with DNA. And so what we do, I'll show you here. So DNA, this is the original DNA that you're used to seeing, right? It's the human DNA, double-stranded, double helix, But here is what we call circular DNA. Bacteria have circular DNA like E. coli, right? And we can use this to our advantage because that means it's, we know it's a fixed size, right? So it has to be a specific size. And we can take things out and put things in to replace them in order to express these proteins that I just talked about. So I know this picture might be a little confusing, but look at the right side. Past the purple point, you're going to see a blue arrow that says inserted gene. So what we do is we use these enzymes, which are in short term, they're just these little molecules that come and cut the DNA at specific parts that we tell them to. So think of it as a rubber band, right? This circle is a rubber band. And you grab your scissors and you cut this rubber band in two different places. And this rubber band is black. So then you take out this slice of the rubber band and then you insert like a green slice. And then you tie the ends of the rubber band and then you make a circular rubber band again, right? You make it complete, but this time the part that you took out is gone and now you have a green part of the rubber band. So that is exactly how these genes work and how we're able to edit with these sophisticated tools in molecular biology and genetics. So that's just kind of how it works. We grab this circular DNA, we take out something that we don't need, and then we put in a sequence, which is just a strand of DNA that encodes for the green glowing protein. And now what we do is we package this into a virus, and then the virus we inject into different neurons, and then it tells the neurons to produce this protein, and then they end up glowing green. So I'll actually show you what it's supposed to look like. Right here, we have some cells that are glowing green, thanks to our understanding of molecular biology. Now, there might be cells around those cells, but we cannot see them. Why? Because genetics is very specific. So we tell this, this virus, hey, inject this genetic sequence that tells only specific neurons to glow green. So you won't see anything else other than the specific neurons that you personally want it glow green. So this is how molecular biology can help projects such as those of Vinit and Jose visualize how these mechanisms work. So as we all know, brain cells, neurons, they talk to each other. Now, sometimes we want to know how this conversation works, right? How the mechanism works. So then we introduce this glowing protein to see how they talk to each other, right? Because you can't record something unless you have the equipment to do something. Like you can't record a conversation without a microphone. This is our microphone. This is how we record how, different conversations happen, types of conversations between these neurons take place. And the advantage of molecular biology is that you can actually look at things either in real time in the animal while it's still alive, or you can see it in cells that have been cultured. So you can see it in the lab in like a dish, or in an animal using a probe that you can insert in the brain, and you can look at these cells in real time. So it's actually pretty exciting just looking at these very small conversations going on, these very small mechanisms, looking at them in real time. So that is basically what molecular biology is and how we use it to our advantage. So I just like to say, before I go to the slide, research, when I first got into college, again, I knew I wanted to be a doctor. I knew I wanted to study neuroscience. I just didn't know how to get around it. Luckily, I was able to apply to a position at Max Planck my second year of college, and thankfully I was accepted, and I worked at the molecular biology core. So a core facility, so within an institute, for example, Max Planck, there are like 9, 10 different labs that each study their own area of interest within neuroscience. And the core facility is just, they kind of freelance and work around with every single one of those labs in order to help them with their projects. So they don't have to do these things themselves, because everybody in these labs needs, fluorescent proteins, all of that. But it's just, more effective to have one facility do everything for them. So going into the lab, I was very nervous. I didn't know all these big words. When I was reading scientific research articles, it's all very intimidating. And, it only took one person to believe in me that I could learn this and I could do this. So through hard work and just resilience, research can be very frustrating at times. And some experiments won't work. Some experiments will just like not work at all, no matter how many times you optimize what you're doing. And it can be very frustrating, but then you learn how to problem solve effectively, and you get into your groove and you stop messing up as much, because I did mess up a lot when I first started, and you shouldn't expect going into research, if you do choose to go into some sort of research related field, that you're going to get everything right on the first try. You're there to learn, your mentors are there to teach you, and I know it can be intimidating. It's a big world. When I was in your shoes not too long ago, I'm only 20 years old, I probably couldn't tell you anything about this or know anything remotely related to my projects in neuroscience. So, it is very exciting that you guys are, getting in this medical academy and in the program so early on, because you have all these, opportunities, and it kind of guides you into what you want to do. So I was very thankful for my time at West Boca and for all my mentors so far in college. And yeah, it's been quite the journey. It's been a journey that I never thought would go the way it has. There's, like Jose, he wanted to be an orthopedic surgeon. Now he's going to be a data scientist. And who knows, my mind might change as well. But so far, I love what I do. I never imagined myself loving what I do, because, I thought of it as like nerd stuff a long time ago. But now I find it that I really enjoy it. So thank you so much again for your time. Our social media is EngageFL if you want to check us out. We also have a podcast that I host. I have different undergraduate students at the Honors College of FAU, and we're going to have all FAU students soon that want to join. It's a quick 15-minute per episode podcast. We talk about their research experience. We're going to have many guests come in from a lot of different backgrounds. So if you're interested in the podcast, please do visit our social media, go on our link tree, and there are all the necessary links. And I don't know, if you want to just check it out, if that's not your thing, that's okay too. But yeah, I'm really excited to share all of this with you, because there's just a lot of amazing things that undergraduate students are doing. And we just want to help bring that to light and create a platform where we can share the amazing things that people are doing.